The sequencing discovery, for which I got the Nobel Prize, was actually an accident. The world has changed profoundly by the DNA sequencing discoveries. And I'm involved now as a consultant in a company that wants to sequence babies. And there's a whole sort of side of should we sequence everybody? And I think ultimately, if we had a rational medical pattern in this country, we would sequence everybody. You would learn something from your sequence that would be of medical use to you.
This week, Jacob Fine spoke with Walter Gilbert, Nobel Prize-winning molecular biologist whose work helped shape modern biology. Gilbert pioneered one of the first practical methods for sequencing DNA, co-founded Biogen, and proposed the influential RNA World Hypothesis, one of the leading ideas for the origins of life. Today, we discuss the discoveries behind these breakthroughs, the future of computational biology, and how, after decades in science, he found a new creative outlet through digital abstract art. Please welcome Walter Gilbert.
The 632 nanometer podcast doesn't have any sponsorships. It's an art project, and we're going to keep it that way. But we wanted to do an unpaid advertisement for Tesla Autopilot. Both of us use it. We've both done probably over 1,000 miles on Autopilot now, and it's just an incredible technology. It's gotten amazing over the last 2 years, and I'm personally very passionate about it because I was hit by somebody who ran a red light, and my pregnant wife was in the car, and it's just something that really shouldn't happen anymore. Like, we have the technology to stop this. If that person was driving a Tesla, it wouldn't have happened. So luckily it all turned out fine.
Driving in Boston is a hell, and I think that definitely helps with finding the right way. It's mind-bending and life-saving technology. I highly recommend.
You have your hands free, your mind free, you can think. Yes, you have to pay attention to the road to an extent, but it really drives itself. But they really improve it all the time. I don't think I've had to take over probably in the last 6 months or something like that. And there's literally all of my driving is on autopilot. Anyway, go out and try this. I think every car should have it, not just Tesla. This software should be in every car on the road.
If you are a fan of the 632 nanometer podcast, you most likely love quantum computers. And now I'm in the lab at Quera Computing, where we build the most advanced neutral atom quantum computers. We have plenty of job openings, and I'm hiring for the position of the quantum machine builder. If you have a relevant skill set and want to contribute to the race for building the first fault-tolerant quantum computer, join us and enjoy the rest of the episode.
I'd met Jim Watson at a party in Cambridge in April 1956. And he got attracted to my wife's ponytail. He came over, she brought him to me, and we fell into conversation and probably talked for a couple of hours at that party and became friends. And then I came back to Harvard as a graduate student. Jim came as assistant professor. He described what was going on in the laboratory to us very often over dinner. And finally, in the spring of 1960, probably in the end of April, he said something very exciting is happening in the laboratory. And he told me they were trying to find messenger RNA, a RNA molecule made by a gene, the bacteria, that would go to the ribosomes, encode a protein, and then after being copied several times into a protein would then be destroyed. And this idea that a gene made a RNA molecule is unstable was the idea of how the information got from the DNA to the factories that made proteins. So I went and watched Jim one afternoon doing an experiment, along with Francois Gros, his collaborator. He gave me 6 papers to read. I went home, I read those papers. That summarized the entire field of knowledge at that time. And the next day I went and joined in doing the experiments. And I found I enjoyed doing the experiments. We worked throughout that summer. We went to a number of meetings. I learned all sorts of molecular biology from people talking at meetings. And we published our results at the end of the year, beginning of 1961.
At this time, you were still working on theoretical physics.
At this time, this was the summer and fall. During this time, I had graduate students in theoretical physics, and during the winter terms, I'd be lecturing in theoretical physics. I'd be teaching electricity and magnetism, general relativity, topics of quantum mechanics, topics like that in theoretical physics. I had, during my career in physics, about 6 PhD students. And but during that time, I found I was more and more interested in biology, and I went back to Jim after doing this messenger RNA work and said I'd like to work in biology, and he gave me a technician and a laboratory to work in, and I began to work on a general question: how does DNA do anything? In which the first answer was, well, it makes a protein, and it does that by making a copy of itself. And the RNA goes to what we knew at that time as the ribosome, and there somehow the RNA is copied effectively in the protein. And the question was, how does that really work? And so I studied that question for a while and published on it. And that led me to a specific discovery that the growing polypeptide chain always ends in a tRNA. And it led me, after doing that, it led me to wonder about what else does DNA do and how does it control? And that led me to the next major work, which was in the middle of the 60s.
And you were interested in basically gene regulation, is how genes are turned on and off.
That's right. The first problem is sort of how does a gene do anything? And the second problem is how are genes turned on and off? At that time, we knew that the genes did not always express fully constantly. The genes in our body are turned on in different tissues to do different things. Each cell of our bodies has roughly the same genes, almost exactly the same complement of genes in it, but the way in which they function is then very, very different in each cell type. So that's the great mystery of differentiation, is how does that happen and how are the individual cells made? So at that time, a crucial finding had been done by the French group, Jacob and Venot, and they showed that you had a gene that metabolized sugar in bacteria, that metabolized the milk sugar, lactose, by a set of proteins. And those proteins were expressed or not expressed. They were not expressed in the bacteria if you didn't provide the sugar. If you provide the sugar, those proteins got turned on and digested the sugar. They wondered how this was done. They thought it was done by having another gene that turned these on, and they named that gene, which they were exploring genetically, the I gene for inducer. And Szilard visited their laboratory at one time and said, why do you think it's an inducer? You should really prove it.
So they postulated something. They theorized that this thing.
Well, they theorized that this gene product actually turned the genes on.
Right.
They had mutations in the gene product that turned genes on. And Szilard said to them, no, you should take 2 copies of the gene in the same bacterium and see which one is dominant. And when they did that experiment, they discovered that the gene product actually turns its target genes off. And the gene mutation that they had thought was the correct one was a lack of that repressor rather than the presence. So they discovered that what we call negative control of genes. And at that time, this seemed to be possibly the dominant paradigm for the field.
Most people thought that the way you did it was with negative control. That was the dominant way.
That's right.
Yeah.
And at that time, people thinking generally thought that would probably turn out to be the dominant thing.
Right.
Today, we know that's only one of many possibilities, and the dominant pattern in our bodies is much more things being turned on than being turned off. Mm. But at that time, the obvious question was, well, what is this material that the gene makes that turns another gene off? The French group thought it was actually an RNA molecule.
Right.
And I tried to identify what it was. And then I and a collaborator, Benno Hill, a postdoc, worked on that problem for several years.
And Benno was he was a research associate?
He was a postdoc of mine. He had done his PhD thesis, and then he came to my laboratory and in fact wanted to work on this problem. And so he and I worked on the problem together. He did an experiment that showed that the product was actually a protein, and ultimately I isolated the protein, which turned out to be an extremely difficult problem because each bacterial cell makes only 10 molecules of this protein. And we had no way of assaying for it except by looking at the way in which it bound to the molecule that turned the genes on. And that turned out to be a difficult problem.
So the problem was the abundance of it was very low.
Well, the abundance was very low, and it had no enzymatic activity. It had no way of detecting it through its action. And so we detected it through seeing how it bound to the sugar. That when the sugar is present, the genes would turn on. And so we could try to look for this binding. But that required that we try to purify and concentrate the proteins inside the bacteria and look into dialysis sacs to see whether a radioactive sugar was being concentrated in the sacs or not. And ultimately that experiment worked. But it turns out, as we discovered later, in the typical way of biology, we were destroying 90% of the protein as we tried to isolate the bacteria, so we were making the problem harder for ourselves than it actually needed to be. Ultimately, though, we could show, yes, we could isolate that material. We could show it was a protein. It bound to the sugar. And then we wanted to know how did that protein work, of which the theory was that it would interact either with the DNA of the gene or the RNA of the gene once the DNA is copied. We thought it was most likely to interact with the DNA of the gene. And we demonstrated that. We actually showed the protein bound to DNA, and then we isolated the DNA fragment that the protein bound to by destroying all the rest of the DNA and having a little 20 base long fragment of DNA bound to that protein. Naturally, that we wanted to know what was the sequence of that DNA.
Yeah. So backtracking a little bit. So you're searching for the repressor. You wanted to find this missing.
That's right. This not missing, but this sort of trace thing. It's controlling a gene, and the first question is what is it? Which the answer is the repressor. How much is there? Well, it's only a little bit there. One of the things we do along the way is make mutants that make more repressor. We made mutants that change the binding of the repressor to the sugar, the inducer.
So one thing that I've been curious about is, you were working in theoretical physics before, and you were particularly working in particle physics. And so did you see any parallels between sort of looking for this particle that was postulated by theory, but did not know or has not been found yet? Did anything from that previous work that you did in physics translate directly to you finding the repressor?
No, nothing translates directly. There is a sense in which the search for new knowledge can be the experimental search for the neutrino, the experimental search for the repressor, the experimental search for messenger RNA. Jim and I would talk about the search for messenger RNA like the search for the neutrino, looking for something that you thought was there, but required difficult experiment to get to, or fantastic experiment to get to. I realized that I was doing this, something profound about, which I think about our nature of education. We often spend 4 or 5 years doing a PhD, and it's a lot of work, and you don't realize why it takes you so long. And what I realized working in the laboratory was that the difficult problem of the PhD is learning how to decide what is true. Learning for yourself how to decide how to do an experiment correctly, how to do a calculation correctly. A great deal of self-learning that goes on. And once that's done, then you can write a PhD. The actual time is spent on this other learning process. It's not actually in the learning of the field. And so you could take that knowledge and do another field, and the actual knowledge scattered for the field is very short. When I began working on the repressor, as I said, I went to a new field. There's not much known about it. I read 6 papers. And
which summarized most of the knowledge at the time.
That was the knowledge. There were a couple of papers on phaged RNA, right. The phage made in RNA. And there were a couple.
And what was one of them, the double helix paper?
No, I knew the double helix
before.
I had heard Jill Crick talk about it when I was a graduate student in England, and that's probably Jim and I probably talked about it extensively at that party originally. So, the double helix was a new fact in biology. It's not all of biology, but although we geneticists think it is all of biology. But I realized after that the actual knowledge to do almost any field only requires a year or so of work. And if it's a new field, it requires very little work.
So you've always picked sort of the right problems to work on, especially finding, going after the repressor, and we'll get to DNA sequencing soon. And I guess, since we're talking about, PhDs and sort of the future generation of scientists, like, is there any advice you'd give to help people pick good problems to work on?
It's actually a very hard question. My advice is almost always to pick problems that you're fascinated by. Don't work on something you think is a good idea that you don't like, but work in a field that you enjoy all the detail of. Francis Crick summarized this by saying you should always work in a field that you like to gossip about.
Yes, the gossip test, he called it.
That's right.
Right.
And it's actually totally right. It's the things that you find fascinating are worth working on, and they keep you motivated. The actual intellectual work can be very hard. It can take you a long time to do things. Mathematicians spend years working on problems. But you have to really enjoy what you're doing. Mm-hmm. As scientists, we generally enjoy the new. I often characterize, I work now in art rather than science, but I see a great similarity in the creative drive in both cases. In the science, our drive is to find something new, but it has to be new and true is generally subject to an experimental test of validity, or for a theorist, subject to the acceptance of the rest of the theoretical community. We tend not to pay attention to history. We tend to like only what has really happened now that no one knows about. And there's a similarity in the art. The driving impulse in the art is to create something that is new. The test is new and beautiful. The beauty needs only to be beauty that I see. If somebody else sees it, that's something extra. But I don't work in the art for the beauty that somebody else sees. I work in it for the beauty that I see. And the novelty of doing something new and different.
Very nice. Very inspiring. So getting back to this, looking for things that are exciting and interesting, you discovered, you isolated the sequence of the repressor, the protein molecule that was the repressor, and that then led you to ask a question. Yeah, go ahead.
Okay, so we isolated the repressor and shown that it bound to the DNA. We then wanted to know what is the DNA it bound to. So we isolated the DNA fragment that the repressor would protect against DNA's digestion of all the DNA. And we could purify that and see it. We have a 20 base long fragment of DNA. And so the obvious question was, well, what is its structure? What is its sequence? We tried to work up DNA sequencing methods, nothing that actually worked at that time. And over the next couple of years, we sequenced that piece of DNA by copying it in RNA and using a whole set of techniques that Fred Sanger had developed for sequencing radioactive RNA. But those techniques involved gigantic tanks with great sheets of paper on which we had electrophoresed fragments of RNA. And the tanks were full of aviation fuel, and we would do high voltage electrophoresis, 10,000 volts on these tanks in a room in which we had, in fact, in case of fire, we had special carbon dioxide extinguishers, which would quench everything in the room. If you were in the room and the extinguishers went off, it was bad luck. And we would have great tanks of aviation fuel all around the halls of the laboratory to provide the But as I said, we worked out the structure. Very difficult. It took us 2 years, and we had the sequence. And the sequence was interesting. It showed a symmetry, and we could realize that the subunits of the protein bound to the DNA, reflecting some symmetrical property in the protein.
Question. So you knew that the protein had to bind in a sequence-specific way.
That's right. Well, we knew the protein bound strongly to one specific sequence in the bacterial DNA. Shortly after that, I forget exactly how it was shown, the protein turns out to have one strong binding site and about 2 minor binding sites in the 3 million bases of bacterial DNA. So it's a pretty unique sequence in the protein.
And so that convinced you, I guess, that it was a good problem to work on to try to then find the sequence and then to develop a...
Well, originally, we wanted the sequence, we wanted to try to understand how the protein bound the DNA. That problem today is solved by X-ray crystallography. There's the problems, the way in which we worked on the problems then, which is the late 60s, early 70s, is in many ways entirely different than the ways in which we get information about the molecule interactions today. Today we get the information from things like cryo-electron microscopy, which gives us, in fact, atomic resolutions of protein complexes. In those days, the X-ray crystallography was still very early, although large numbers of protein structures have been known or had been worked out through the X-ray crystallography. But something like what is the nature of the repressor binding DNA would have been impossible to tackle that way. The sequencing discovery, for which I got the Nobel Prize, was actually an accident. I had found the sequence of the Lac operator. About a year later, a Russian, Andrei Mirzabekov, did a tour of America, visiting laboratories. He led out of Russia at that time. And he came to the laboratory and he said, "Wally, you must do an experiment in which you see how the repressor interacts with DNA." Mirzabekov had been working with agents that bound to DNA, and he wanted me to use an agent called dimethyl sulfate, which modifies the DNA, and asked, did the repressor block that modification? And that would show us, did the repressor touch the adenines in the minor groove, or did it touch the guanines in the major groove? And that would be a very interesting experiment, but I couldn't think of any way of doing it. 6 months later, as he's about to leave America, he comes back and we have lunch together again. And he says, you really should do this experiment. And that time I thought of a way of doing the experiment, which was to take a fragment of DNA and put the repressor on it and modify the guanines or the adenines with this reagent. And I realized that I could break the DNA at the position of the modification and use the length of the DNA from some point of radioactive labeling to identify where the guanines and adenines were. I, by that time, knew the sequence of the entire region. Another group had worked it out by copying it to RNA. And I myself wasn't interested in sequences. I knew all the relevant sequences of the genes I'd been interested in. But I did that experiment, trying to ask, can I detect how the repressor touches DNA? And the second time I did it, it worked. The first time it failed completely. Second time it worked like a charm. And I got a pattern of bands on the photograph from having separated molecules by size on the electrophoretic gel. And they could see that the repressor clearly prevented the bands appearing in the tube in which it had been in, and the bands appeared in the other tube. But the pattern was so discrete that not only could it say here the repressor is touching a guanine at position 25 and it's touching an adenine at position 30, but I could look at the pattern and say, oh my God, it's actually a sequencing method, because I get a display of the guanines from positions 10 to positions 50 or 60, and I can see the all the guanines and all the adenines just laid out as a ladder on a gel. So I said to myself, this is going to be a sequencing method. And I immediately tried to see if I could sequence the other strand, which I did, but we never published. And then my technician, Alan Maxam, began to look for the rest of the method. And it took us about 9 months to find 2 more chemical reactions that then would distinguish the C's and the T's. And we published a paper using the method and gave the method away at a Gordon conference, I think the summer of 1976. And everybody who heard came to they gave the method away in mimeographed instructions. People who couldn't follow the instructions came to the laboratory, learned how to do it, went home and sequenced, and everybody began to sequence genes. Fred Sanger discovered simultaneously in 1976 another method that displays similarly the sequence of genes called the dideoxy method. And both these methods got used around the world to sequence genes. People began, in America, used our method mostly, and Europe used Fred's. By 1980, 1 million bases of DNA had been sequenced. And people, the world was now one in which if you wanted to know what your gene was, you would sequence it. By 1977, I think, a graduate student sequenced 1,500 bases of DNA for his thesis. He sequenced, in fact, the repressor gene, discovered the protein structure was wrong. It had been worked out with great difficulty by that time, but it was actually had a mistake in it. Next graduate student sequenced 4,300 bases of DNA, sequenced the entire plasmid, and we stopped giving PhDs for sequencing. It was now too easy.
So what was your reaction, when you first saw that gel and you saw the banding patterns being so distinct, looking like, you got the sequence?
That's right. You could read the sequence. I mean, something that had taken years, oh my God, look at that. There's a display that shows me that there are 2 A's at position 31 and 32, and I can see a guanine here, and I can see the adenines there. I can just look at it and see the sequence. Fantastic moment.
Yeah.
Because it meant, the development of the sequencing meant that in an afternoon you could do first hundreds and thousands of bases.
Scaling by like Moore's law almost.
Well, it turns out, you see, now let's step back a moment. We're going from 20 bases in 2 years, a base a month, essentially impossible to work out a gene, to, hundreds of bases in an afternoon. Easy to work out, 1,000 bases, 4,000 bases, 10,000 bases, perfectly straightforward. Genes begin to be published. We publish the first. We debate, of course, how should we write the gene structure that we want to publish? Should we write it A phosphate, G phosphate? Should we put in the sugars or phosphates? How should we write it? We make a decision about how you should write the DNA sequence, and it finally gets the way we do it these days.
5 to 3 with just the letters.
Just the letters.
Yeah.
And it's a fabulous moment. It's both the moment you see that first gel. You can see that the technique is there. Of course, you see, to get the rest of the chemistry working is not obvious. I go to a very good chemist as a friend and say, how can I distinguish the bases chemically? And he says, you can't. They're too similar.
Right. And as I understand, that was one of the reasons why it took longer to develop DNA sequencing than it was to develop protein sequencing, because it was harder. It was easier to tell apart individual amino acids than it was to tell apart bases. And
that's right.
Your chemical modification method, which involved cleavage, basically you added specificity to the sequencing method because of the modifications.
That's right. The conceptual trick in the method is that by the modification is done at a very low concentration of reagent, in which you're modifying 1 out of 1,000 bases, 1,000 versions of that base.
Right.
And so this is a condition with highest specificity.
Mm-hmm.
Then once we've modified, we strip the base out, and then we cleave the DNA with a harsh agent. Harsh agent wouldn't distinguish, but it's got now a target, just a free sugar, and you can easily cleave the DNA. If you try to modify and cleave it one go, you lose everything. So the whole trick is to have a specific reaction that's used in its greatest domain of specificity, and then a harsh reaction that then gives you the fragments you want. We quickly, as I said, everybody begins to sequence. People flock to the laboratory, learn sequencing, go home and sequence genes right and left. The sequencing begins to be very prominent. Everybody recognizes that the sequencing of genes is something really important. So we get all sorts of prizes in the late 70s, 75, 76, 77, 78. We're, by that time, deep in another problem, which is how to make proteins in bacteria, human proteins in bacteria. But by the time of the Nobel Prize in 1980, we've won a number of the prizes, Fred Sanger and I, on the way up. And by that time, about 1 million bases of DNA have been sequenced around the world. Databases of DNA have come in, and I can look at those databases and realize that the sequencing around the world is following Moore's law. It's going up a factor of 2 every 18 months, factor of 10 every 5 years. There it's only going from 100,000 to 1 million, but I can see it's going to be 10 million in the next 5 years and 100 million by the 5 years after that.
So at what point did you realize we might then be able to sequence the entire human genome?
That's what in 1985.
Right.
I could see the sequencing increasing all the way across this period. And between 80 and 85, I was involved in a company, and actually from 81 to 85, I was running the company. And we were trying to make human interferon in bacteria for medical treatment.
This was Biogen?
This is Biogen. And we started Biogen. We can go into that. It's a separate set of stories.
Separate story. Yeah.
But the 85, I had stopped doing Biogen, and I was trying to decide whether to go back to Harvard or go to MIT. And I get invited to a meeting that Bob Sinsheimer organizes in California. And the title of the meeting is Sequencing the Human Genome. And he called a meeting to sort of have people discuss whether it was possible. And I go to the meeting thinking, that's the silliest thing I ever heard of. And I listen to the talks of the meeting and very quickly become convinced that it's a perfectly doable problem. It's clearly a question of industrial scale. I realize that somebody using our methods or Sanger's methods, working hard, could do about 100,000 bases a year, and therefore 10 people could do 1 million bases a year, and 100 people could do 10 million. You could easily imagine the scale up. And I knew both that as you scale the industrial process up, the costs drop and the technology constantly improves. So I could realize that in principle, we could start at one end of chromosome and just go to the other, and we had a linear way of putting the sequence together. And that if we once had that sequence, it would be a tremendously useful resource. And it would change the way we thought about the way in which we did all of our gene research. So on the way back from the meeting, I wrote an essay in the plane that I sent to Bob saying he should set up a institute and do it just locally. 300 people working for 30 years could do the whole thing at a cost of roughly $3 billion. And I went to the Gordon Conference and talked up the idea, again putting a price tag of about $3 billion. I thought it would cost of the order of $1 a base to do. And that talk got people enthusiastic, some people enthusiastic, other people hated the idea. And the Department of Energy got I began to get interested in the idea. We had meetings at Cold Spring Harbor. Again, I talked it up, and One type of person would get enthused by the idea of creating the knowledge, and another would be upset because it wasn't the research science they're used to. It was an industrial scale production, and which the interesting result is not the sequence, but it was the knowledge that would come from the sequence once you had it and could produce it. But the sequencing itself was basically a technician's problem.
And in many ways, this was really a scaling problem. The basic technology was developed.
It was purely a scaling problem.
Right.
And I knew even the basic technology done at appropriate scale could do this.
Right.
In reality, machines came in. The technology had already improved by the time we did the first sequence, which was by about 2000. And the technology has steadily improved since then. We've gone through multiple generations of better and better sequencers. They still come out with better sequencers. The first genome actually took about 5 years to do in that period, mainly because our programs got deflected by people trying to make better methods rather than just use the methods that were there. Once they realized that you had a perfectly good method, it was purely a question of scale. Large laboratories began to set up and do it. And the first genome was done that way. But by now, as I said, the first genome was done in sort of a 3 to 5 year period. By now, we can do a genome in about half an hour with the current machines.
So by accumulating all this data, you were almost creating because PhDs sort of in the 80s were focusing on cloning and sequencing a gene. They were doing one gene at a time. You were almost, by doing this, you're almost putting out of business a lot of the single gene focused people. So what are your thoughts on that?
You're quite right. I used to go around giving seminars in the 85 to 90 period. I'd go around giving seminars I'd give seminars describing aspects of the technology, just trying to think of the real timing. At times I would give a seminar on the sequencing late 70s. I give a seminar in which I'd talk about the Lac operator, I'd talk about the sequencing method, and then I'd talk about actually putting DNA pieces together and making novel genes in bacteria. And once the idea of the human genome came, I began, as I would give seminars, I'd say to the graduate students, we're going to break your rice bowl. You think the world is one in which for a PhD you identify a gene and work out its sequence. And the world is going to be one in which you can look up all these sequences. You'll have me doing something totally different for your PhDs. The world is going to change entirely.
So you in many ways predicted the rise of computational biology by this, that you could query.
Well, completely, because in fact, we didn't know the methods. I mean, I was thinking of BLAST, and these things began to be developed in the, actually 90s. It's that period. BLAST is probably 86 or 87. So it's right in there. I'm just trying to think when the real comparisons of fast comparisons of DNA sequence came in. But the original idea behind the genome, you see, was of course, it's what we would now call computational biology. It really was the idea you would have the sequence and you would look up in it all the relevant genes. Now, at that moment, as I'm saying this in 85, I'm estimating that there are 100,000 genes in the human genome, that the gene is, by that time we know about splicing, the gene is probably 30,000 bases long, 100,000 genes is 3 billion bases, and that's roughly the right number. As the sequence gets done, we discover first that estimate is wrong. There seem to be only about 20,000 protein coding genes. Everybody goes through, the rest of the DNA is just junk. We now are in a different world in which we realize the rest of the DNA, all sorts of the DNA is doing things. RNA molecules are being made all over the DNA and doing all sorts of interesting genetic control, from long RNA molecules that do things like wrap up the X chromosome to short RNA molecules that
it's interesting because it's almost like regulatory RNA, the idea that, Monod and Jacob predict their
Well, that's
original
right.
idea
They
of
thought it was a regulatory RNA
RNA.
molecule, and that
Yeah.
sort of disappeared for quite a while.
Right.
Now it's finally come back.
Yes.
I mean, the microRNAs, the small interfering RNAs, there's all sorts of regulatory RNAs that work in all sorts of interesting ways.
The modern RNA world in some sense.
Yeah.
So we could also, there's lots we could talk about. We could get into the RNA world, to the origin of life. Maybe we can go there. So, going back to the 80s, late 70s sequencing is
That's right.
sequencing. How does that then lead to you starting to think about how life began, and in relation to some of the discoveries that were made in the early 80s?
I was just trying to think of. The interesting discoveries of the late 70s. We have the DNA sequencing. We have the cloning and the butt-end ligation that enables you to move promoters around, try to make proteins deliberately. So by the Nobel Prize, by the way, is for the recombinant DNA techniques.
Right.
That was the nature of the prize that year. So they gave half to Paul Berg,
Yes. standing
in for everybody who moved DNA fragments around from one cell to another, and they gave half to Fred and myself for deciphering DNA. Didn't matter if you moved it around, you couldn't figure out what it was. It wasn't as useful as understanding everything. The prize is only given to 3 people, so that it's unfortunate that the rest of the people who really discovered how to move DNA around did not get rewarded. In the early 80s, I was very much involved in Biogen, and the problems then were actually the technical ones. What are useful genes to try to express in useful human genes to express in bacteria? How do we make the human protein in large amounts to serve as for medical treatment? We actually made interferon and brought it to shearing plow, and the first interferon that went to patients was made by Charles in 30,000 liter fermenters that we hired in Austria. So I remember it. And Charles purifies the protein, and then he, being a traditional doctor, he injects himself. The large dose of the protein, wouldn't give it to anybody. He's a doctor of the old school, scientist of the old school. He would not give something to a patient that he hadn't tested on himself and knew it was safe. So he shot himself up 100,000 units and found himself lying on the floor and gets up, tremendous headache and everything else, but recovers and decides, okay, I guess it can be used.
Wow.
He gives it to shearing, and they try to use it in a variety of things. It turns out to be specific to cure hairy cell leukemia. And it first gets a license for use for that. But then interferon has an interesting career. It doesn't get used that much in virus treatments, except it gets used to treat hepatitis B and becomes a major drug, major pharmaceutical mostly in Japan and China and Western Europe, places in which hepatitis B is much more prevalent than this country. But that I've sort of wandered astray. Back into 85, there's a discovery of RNA enzymes by Czech and
Sid Altman.
And I think I must read Czech's paper. I'm not. And begin to wonder, well, if there's RNA enzymes, maybe RNA can synthesize RNA, and that would be a sort of simple beginning of the world. And so I get enthusiastic about the idea, begin to talk it up, write a paper on that called the RNA World, which sort of tries to pursue that idea.
It's a one-page paper that said almost everything that needed to be said at that time, and was pretty inspiring to many people. So were you particularly interested in the origin of life, or was it that you saw the question?
I'm interested in all sorts of things, you see.
Yeah,
so
yeah.
It's not that I'm particularly interested in the origin of life. I think the origin of life is one of the great questions. How the brain works is also a great question. How you build the body is also a great question.
Right.
But if I were starting out today, I'd probably work on how the brain works. But
Yeah.
at that time, and actually what I worked on changes from time to time. I don't feel that I have to work on the same idea all the way through. So that I think, the RNA could be an enzyme. There may be many RNA enzymes hidden away in the cell. Actually, when I wrote the paper, I expected a large number of RNA enzymes to appear as one began to go further into the RNA, in the metabolism of the cell. The paper uses the Czech example, and then uses a number of examples of protein enzymes that are essentially of RNA cofactors that do the enzymology. I was, of course, delighted by the discovery that the ribosome
is a ribozyme.
Is that the crucial site that puts peptide bonds together is actually an RNA enzyme. And so the RNA world is the idea that you begin with RNA molecules that copy themselves into other RNA, and therefore that you can begin an evolution truly with one molecular type, and the molecule can then mutate and modify and copy better, and evolution can develop.
So the key question in The Origin of Life with respect to the central dogmas, you have DNA, RNA, and protein, right? And so what I guess, could you explain to us maybe like what the RNA world solves about that? Because it seems yeah.
Well, actually, let's go back. In most of life as we know it now, DNA makes RNA, and the RNA then DNA is copied in the RNA, and the RNA then is translated into protein by its own mechanism. And you have the problem of, in the origin of life, is what is the simplest possible system? Mm-hmm. There's a whole group of chemists who always like the idea that you made peptides, and you made proteins, and proteins can be enzymes. Isn't that wonderful? The only drawback with that is that the proteins as enzymes are generally three-dimensional structures rather than structures that carry the information to make themselves. The moment DNA could be a starting material, but DNA requires proteins to make it. It's hard to make. RNA seems to require proteins to make it. This is back earlier. The moment somebody discovers an RNA enzyme, you could suddenly say to yourself, well, an RNA enzyme could copy RNA and make more RNA. We now have a much simpler system. And if we now go back and look at the system that we want, in my view, is a system that can undergo Darwinian evolution. Problem with getting life started is to get something which can evolve. And Darwinian evolution works by having random changes change the way in which the organism is growing, and those random changes which improve the growth ultimately get selected for. And in my view, that's the critical idea behind life. Not energy and not energy, but replication with error and evolution.
Yes.
So that the moment you see here's an RNA enzyme, well, I could imagine other RNA enzymes. I could imagine RNA being copied in the RNA, right, mutating for better copy, doing other things. And then as you look at, if you then step back one stage and say, well, where does DNA come from? The actual biochemistry of DNA runs through RNA intermediates, so that DNA does not look like a primitive material. It looks like a much more sophisticated material. It's not made automatically from DNA intermediates. It's actually made in a much more complicated fashion. And the protein enzymes involve RNA components that do a crucial part of the enzymology. So back in 1985, when I wrote that paper, I think it's
86. It was published 86. I think,
okay,
yeah,
it's probably 86. I expect that there will be many, many more RNA enzymes seen, that RNA enzymes are clearly a simpler form. It's sort of almost automatic to say, well, the simplest way of getting life started would be RNA molecules copying themselves and mutating the better and better copying patterns. Now, unfortunately, in the 40 years since then,
We haven't really found so many.
we haven't found RNA molecules that fully copy them. We've made
We can make them.
we've made some interesting molecules that do copy quite a bit, but not ones that are fully self-replicating yet.
And not ones that are naturally occurring.
Well, it's not clear that any of the original RNA molecule would be naturally occurring.
But if someone were to find such a thing, that would be quite significant.
If you found a naturally occurring one, that would be very nice.
Did you think that someone would find one day when you were working on this? Did you predict that maybe someone would in fact
No,
isolate
I thought it would be done synthetically.
Okay.
I didn't expect
Anyone to find it.
I didn't expect it sort of to still exist
But
in a sort of full form.
now with bioinformatics, it seems that, with all these massive sequencing databases that we have, it seems like a good idea for a project might be to like search all those databases and try to find anything that looks somewhat like an RNA self-replicating enzyme. But what do you think? Would that be worth doing with someone out there?
I don't think you know how to characterize a self-replicating enzyme. That's the problem is you might search the databases of RNA. Well, they're not so much databases of RNA. They're databases of DNA sequence.
Right.
The major database we have. There's just been an amusing paper claiming that by searching databases of bacterial sequences, they try to find conserved patterns that might be bacterial genes to prevent virus infection. And there's been a number of searches, and in fact, the groups doing those searches ended up with sort of 100 groups of genes that might be doing something, none of which in that groups were things like CRISPR and things that were known antiviral components, but many that aren't. And one of them has just been deciphered and involves a protein encoding an RNA. Protein sequence is actually, they claim, is actually used to put together, to dictate the RNA sequence that binds to, or that it creates. So it's possible, it's not, would never, in thinking back on the time, one would not have suggested something like that. Of course, the body of knowledge we have now is very, very different.
And you sort of foresaw this, when sequences started to accumulate. There was a paper you wrote in the early 90s about, that maybe biologists should start to become more computer literate and do sort of things like this.
That's right. There are two. Yeah, I'm just trying to think what I did. I wrote a paper called Molecular Biology Instead, Long Live Molecular Biology, which was in fact the sort of idea that the sequences would show us a lot of things. I wrote an essay about doing the human genome, which probably also has some of those ideas. And again, the ideas are ultimately ones. We should be careful because the world we live in is so computerized that it's hard to think back to the periods before it. It's the power of the cell phone I carry in my pocket is unbelievable compared to the first computers. I played with my first computer in 1976. It would have been a PDP-8, and you sort of programmed it by throwing switches and actually putting the program in one command at a time. Then the first computer we had in the laboratory, the Z80, big flop. First time I saw a 500 kilobyte disk, half a meg, it was a fantastic thing. I mean, when one isn't able at that time to predict the future in the way we see it now. I mean, now we see a level of computer analysis that was unforeseeable.
Yeah.
Other than, something might emerge, but they really detail that it emerges.
So a lot of the comparative genomics work, for instance, has revealed quite a lot using computational methods. And I'm also curious, I mean, I know like Sidney Brenner, I think taught himself how to code at one point. And he did think a lot about computational concepts in biology as being maybe useful for a language to understand biology. Because, you could do a bunch of experiments ad infinitum, but really if there's not a core theory underlying biology,
Well,
it's kind of
that's
hard
right.
to...
Well, it's both.
So what are your thoughts on that?
Well, we still don't have a core theory underlying biology. So I'm thinking that there's it's sort of a moment in which we're working toward the first human genome. When we have a human genome, do we think of having 1 billion human genomes? I mean, we're about to move into a world in which our computers will be able to analyze 10,000, 100,000 human genomes, even 1 billion human genomes. We begin to do other genomes, and we have sort of single examples. Then we begin to have multiple examples. We have bacterial genomes because Greg Vedder sort of sailed around the ocean, sucking up bacteria and sequencing them randomly and trying to put things together. It's an amazing, it's an amazing world. You don't actually foresee it in quite that, the form that you're asking, do I foresee, oh my God, the computer use? It's like, I still remember the first iPhones, the world changes. The world changed with the first flip phones, and then it changed when the iPhones came in, and suddenly you could access information. And with Google, first with Google and the ability to search bases around the world, and we're now in a world in which is getting digested by AI.
Yeah, that's something else we could probably talk about. But I mean, also in that article, there's sort of an idea of, biologists have most like biology education training historically emphasizes knowledge of doing experiments, maybe some very simple data analysis, at best maybe first year calculus, but there isn't really much emphasis currently on rigor in math and theory, although that's changing. So
that's
do you think that would benefit? Would that benefit? Should more biologists learn math? Because you often hear people say, "Oh, I'm a biologist. I don't need to know this matrix multiplication, or I don't need to know that." But now you see more of that being needed. So
right.
What are your thoughts on it?
It's not quite coming in. I mean, there is the computational biology. You're doing sort of computational biology, and that probably doesn't involve fancy matrix multiplication, but
it does a little bit, yeah.
It does. The machines are doing it for you.
That's right. Yeah, there's Python packages.
That's right. And no one's doing it by hand anymore,
No.
so it's not. It's not clear what people learn. Well, it's actually the whole learning process is shifting under our feet as we go forward. Yes, it's good to know some mathematics.
I think so. I
As
know
a theoretical physicist, I always thought it was good to know lots of mathematics. On the other hand, there's lots of pure mathematics that I don't know at all.
there's a quote I think by Sidney Brenner who it was in an interview. He was like, someone asked him, I want to do biology, what math should I learn? And Sidney Brenner said, you have to know how to count to 10 or something very simple like that. And that's not true anymore, but maybe in those days you could do a lot of stuff without much math. But now,
well, that's right. Of course, you had to do, you always had statistics.
Yeah.
And therefore you had to know things like the Poisson distribution, and therefore you had to know enough math to understand these things.
So going back to thinking about gene regulation in the 70s, you have the famous Sharp-Roberts discoveries of splicing. Was there anything in your work that sort of maybe you were wondering about that was perhaps made more clear by these other discoveries?
Yes, there was a whole component in the work we did. After we discovered the DNA sequencing, met Susumu Tonegawa at a Gordon conference. We shared a room together, and he had just cloned the light chain of the kappa light chain of the immunoglobulin. He was trying to, in fact, he hoped to ultimately see the entire gene. He had a clone of the light chain, and learning of the DNA sequencing, he then came to the laboratory, both to learn how to sequence and to get the sequence of the light chain. And basically, my technician, Alan Maxam, did the sequencing. And in that sequence, which was just of the V region, we could see a sequence that corresponded to a hydrophobic leader sequence of the protein, followed by a gap of about 15 bases, as I remember it. And then the sequence then continued into the light chain. We didn't know what to make of that. We expected just the sequence of a protein to be continuous in the DNA. I then went to the Cold Spring Harbor meeting that summer, and I know this sequence. At the Cold Spring Harbor meeting, Phil Sharp and Rich Roberts both talk about their discoveries of RNA splicing, which they've now seen in the adenovirus sequence. This is an amazing discovery of excess regions in the message which are not there in the final express message. I then travel in France and Switzerland that summer, and in conversations in Geneva, I'm talking to a yeast scientist about problems in yeast, which also then are strange genetic problems that are resolved by the idea that there's some sort of splicing that expresses the protein. The positions of mutants and the position of the protein don't seem to be quite what you would expect on the DNA.
So did most people at this time think genes were continuous?
Well, all people at this time thought genes were continuous. Right. The splicing showed that genes were not continuous. And
which won the Nobel Prize.
Yes, the discovery of splicing won the Nobel Prize. Now that summer, we were both writing a paper. I'm writing a paper with Susumu on the sequence of the light chain of the B region of the kappa light chain, and it comes to me that maybe the splicing and the fact that the genes are separate, have parts that are separate along the DNA, has a evolutionary explanation. And so I then write a paper called Why Gene in Pieces, which argues that by taking regions of protein and pulling them apart along the DNA, you would increase the recombination between those, enable those regions to be shuffled independently at a much, much higher rate than the illegitimate recombination that you might have thought would be used to reassort portions of protein into other proteins.
So instead of there as a mechanism to introduce novelty in evolution,
well, that's
it was
right.
a lot cheaper to innovate at the RNA level rather than the DNA.
Well, it's not that you're innovating. Well, you are. In this case, you're innovating at the DNA level. You're making a novel gene. Well, I'm making a novel gene by getting parts of two genes to interact along the DNA. And if the two genes are very close to each other, it's very unlikely that two regions of a gene are very close together. It's very unlikely it could be separated by recombination. Right. If they're 1,000 bases apart, it becomes very easy for them to be separated by recombination. And so this is the idea that we call exon shuffling, the idea that a protein is made up of a series of units which could be shuffled and redealt into other patterns in other proteins. And I give them the names of exons and introns, exons being the region of a gene that expressed, introns being a region of a gene that were internal.
Yeah. Was there, out of curiosity, was there anything in particle physics that influenced you to
I
say
don't,
intron and exon?
I don't think so. It may, although it may have been. I thought it was a natural name for a thing. I realize so we use the word ohm very often in biology, but we didn't use the word on particularly.
Right.
They come back to me in some form.
But now more people have talked about ons since you coined the terms exon and intron. You hear about like regulon and other sorts of things like that, replicons and things like that. So it's interesting to think of how the language changes.
That's right. So in any case, so I began talking up this idea, thinking of it as the origin of eukaryotic genes, and thinking of I even used to give fun seminars saying, well, the Cambrian explosion is really the moment in which we create eukaryotic genes and reassort the pieces, and as gene structures became available, one saw more and more of this. The two amusing stories out of this. Susumu Todogawa had not yet cloned a functional immunoglobulin gene. And again, that summer, we're probably meeting in Switzerland somewhere, and we bet. He bet that when they put the gene together, this extra little region in between the leader sequence and the V region would be lost. And I bet the other side of that no, this region would be there, and there'd be other intervening regions, other introns in the final gene. And we bet a case of champagne. And then he cloned the gene, and I won. So he sent me the case of champagne.
What year?
I was about to tell another story. That's right.
Sorry, around when was this? This was like the late.
This is already around. This must be 77, 78. That's probably 78.
This was when. So the Y genes in pieces was around that time.
That's right. The Genes in Pieces is written, I think, probably published in 77. That's why I can't remember. I have to look it up.
And when was it that it became clear that splicing was a very powerful way or a very useful way to approach mammalian gene regulation? Because to me, it seems like that was really a key step that paved the way to...
That's actually a much later element.
Okay.
It becomes clear as one sequences mammalian genes. I mean, both at that moment, you run across things like a 50 base long intron in Drosophila that's actually used the timing mechanism for gene expression. It takes so long to get through that intron that in the early development, genes are expressed at just the right time. The evolutionary idea that I suggested was the one that suggested we should see the evolutionary reuse of protein pieces. Ford Doolittle was at that time doing a sabbatical at Harvard.
Ford was an evolutionary biologist.
He was an evolutionary biologist, and he sort of looked at the argument and sort of said, maybe the genes always were in pieces. And so he first expressed the idea that the genes might always have been in pieces.
This is introns early.
That's the introns early. I thought that was an excellent idea, and became a strong proponent of the idea that maybe the very first genes had an intron exon structure, because you want to assemble the gene from small polypeptide pieces whose sequences could then arise in some natural way, and then the larger protein structures could be made up of small pieces put together by splicing. And again, the shuffling of the pieces would be useful behind genes. This argument, I was very positive about this argument. I probably still think it's probably right somehow deep down underneath the introns early, but one struggles with that.
And did that influence your thinking about the origins of the RNA world in particular?
No, it's really after the RNA world ideas. There were RNA world ideas that RNA enzymes and RNA early in evolution.
The origins of protein
I guess I'm just trying to think of the timing of things. It's still hard to recollect. The RNA World, I guess, is 85.
86. Yeah.
86. So that's right. So this is the early, James Early is now is back in 77, 78. At the Nobel Prize, I actually probably talk about early introns in my Nobel Prize lecture.
Okay. Wow, I didn't know that. I'll check that out.
Yeah, you should look at that.
I'll check it out.
It's probably amusing. That's all on the Nobel website.
Great. Well, I guess in the final stage of the RNA world in your paper, you talk about the origins of translation, because really the origins of protein synthesis is one of the other key things that the RNA world explains, is that translation is like the oldest thing. It's older than DNA replication and RNA transcription, right? From a DNA template.
That's right. But it's after RNA transcription from RNA templates.
Yes. The RNA, RNA dependent.
That's right. So first you make RNA enzymes, and they do all sorts of things.
Yeah.
And then you begin to make polypeptide fragments, enhancing the RNA enzymes, and then you finally begin to make polypeptide enzymes.
So then at what point, so then it seems that then in the very simple origins of translation, it was the simple proteins that were first made, you would have predicted that exon shuffling would then explain how domains would rapidly accumulate.
How complex domains could be put together.
Yeah. And then, I've always found it interesting that, DNA replication begins with an RNA primer, which is then removed.
Yep.
And to me, when I see that, I think, that seems like another possible relic of the RNA world.
That's right. No, it is. That's right.
And that's what you think. Okay.
Absolutely.
So that sort of thing. I remember.
That's right. If it was known, I would have cited it in the RNA World paper. So I don't remember at this interval whether it was known then or became known shortly after.
Because I remember asking my biology teacher, one of my biology teachers, why are there RNA primers? What's the point of making a primer and then removing it? And she was just like, oh, because RNA polymerase needs a 2' or it was just some explanation that didn't have anything to do with the origin of life. But then when you think about the RNA world, it just made that very clear that it's the RNA relic. It's a relic of the past. So I find that very interesting.
Let's talk about the origins of Biogen.
Yeah. So I guess you guys were both co-founders of Biogen. So how did that start?
Actually, Biogen gets started by two wandering venture capitalists, and they run into Genentech, which just begun in probably 76. And they hire Phil Sharp as an advisor and take him to see Genentech, and he looks at it and he says, that's a good idea, you should invest in it. So they make an investment in Genentech. And they look at Genentech and they think, yeah, that's interesting. Maybe there's some interesting other science things that can be done. And they talk to Phil. Phil brings them to me. They talk to me. And they go travel around Europe talking to scientists. And the idea that's in the back of their minds is, well, Genentech's interesting. Maybe there's room in the world for another biotech company. There'll be room for a second one in Europe. So let's set up a biotech company in Europe. And they identify 10 European scientists who might be interested. There's Phil Sharp and myself. And they invite us all to come to a meeting and discuss, is such an idea of setting up a company in Europe in this new field of developing proteins and bacteria interesting. At that time, I was trying to make, the recombinant DNA technologies had moved to the point where one could begin to think of making human proteins and bacteria. And I actually had a set of such experiments going on in the laboratory. I had a graduate student whose project was to make E. coli bleed by putting a globin sequence in the bacteria. He never worked out, unfortunately. And then we had a project to try to make insulin in the bacteria right across that time. And in 1978, we actually managed to make rat insulin and rat pro-insulin in bacteria. and sort of one of the first mammalian proteins actually made in bacteria. So we were deep in that sort of problem.
Trying to make human
And so we were very interested in the idea, can you make useful proteins in bacteria?
Right.
And, the idea of human proteins is, can you make proteins that could be used therapeutically? Otherwise unobtainable human proteins. So we had a meeting, the venture capitalists had a meeting in Europe of 12 scientists, the venture capitalists.
Where? Sorry, New York?
In Europe.
In Europe.
No, we
Yes,
met,
right.
we met,
Okay.
met in Geneva, and then we had a series of meetings in Europe. Most of the scientists were Europeans who just filled my show, from America. And as I said, the idea behind the company was originally to be a patent-holding company in the Dutch Antilles that owned structures in which, the functional company would be in Switzerland. And after about 3 meetings, they had raised a little money. They had raised $75,000, of which only $37,000 was actually in cash, and the other $37,000 was in credit from International Nickel. And they invited the scientists to join in starting the company, and we all met as a group of scientists. And we all discovered, we thought, yes, the company might be useful. That we all thought the technology was very powerful, could make useful proteins, useful things in humans. We were very distrustful of the venture capitalists. So we structured the company. We said, we go with a company, venture capitalists outside the room at this time. We go to the company, but we'll constitute a sort of scientific board of the company, and we'll control the stock issuance and control the budget. And the original idea was we do research in the laboratories of the scientific board members and make discoveries, patent them, and sell rights to the patents. So our 2 venture capitalists started to find all the scientists who've gone with the company, and they've elected me chair of this board of scientists. And the board of scientists also has a group of scientists. We also demand to have a third of the board of directors. So we then constitute a third of the board of directors, and I'm elected sort of vice chairman. Mm-hmm. And the venture capitalists provide the other part of the board of directors. And we start doing research, and we start also trying to raise money.
And you still had a lab
I
was doing academic work.
I had a laboratory at Harvard doing academic work during this time. This is 1978. And
how did you balance this stuff at the beginning? You were doing a lot of things, so how did you?
Well, the company sort of say to the Harvard, well, I'm going to spend 1 day a month, or maybe 1 day a week, with a line clear, not too much time on the company.
Right.
We have the company pay for the telephone line in my office, so I have a company line. I'm not using grant money or anything to support the company. The company originally was setting up in Europe. It's trying to decide what to do. Charles Weissman proposes we sort of pull around. Charles Weissman proposes work on interferon, alpha interferon. I propose work on insulin and human serum albumin, I think. Ken Murray proposes to work on hepatitis B. There's a variety. I can't remember.
And what was the overarching goal of the research program at Biogen? It was at this time.
Well, the initial idea is let's make useful human proteins in bacteria. Uh-huh. And
did you interact?
For example, it's the interferon.
Yeah.
What Charles is working on, human interferon. Human interferon is being used as a treatment, antiviral treatment, experimentally, and anti-cancer treatment. At that time, we thought of it as a useful antiviral, but it becomes very prominent as an anti-cancer treatment just at the time it gets cloned. So Biogen gets started in 78. At the end of 79, Charles actually manages to clone human interferon in his laboratory. The company is actually enabling Charles to work because at that time he could not have worked with human cells directly in Switzerland. And he needed to get a supply of human cells that are making interferon to try to get the message that made interferon. So he flies to Iceland to a laboratory a center that actually makes human interferon, gets cells from there, flies to Germany where they have laboratories in which he can then work with the human cells and try to isolate and clone a gene for the interferon, takes the material finally to Switzerland and manages to actually express it in Switzerland. And all of that sort of interaction is being paid for by the company.
Mm. Which is mostly this was still VC money.
Well, at this point, it's VC money. I begin to try to raise money along with the two venture capitalists, and we travel around. We talk to companies who are trying to raise money. We then talk to Shearing Plough, and we get them to invest $8 million in the company. It's the previous investor said it's $75,000.
And $8 million today, that's a lot.
It's a lot.
Yeah.
75,000 was, quite a bit of money back then, but not as. We talked them into investing $8 million at a company valuation of $32 million.
Wow.
And International Nickel, which is the home of the venture capitalists, then puts in another $1 million and a quarter. And with that money, we plan to open laboratories in Europe. Before that, we're doing research in Charles' laboratory. We're doing research in laboratories around. I'm trying to remember the exact timing of things. The investment must be in 79. We must have that money by the time Charles, because what we had promised Shearing in order to get that investment was that we'd make 3 products for them, one of which was going to be alpha interferon.
Mm-hmm.
And that was the only successful one. I think we promised 2 others we probably never delivered on. But so the alpha interferon was then, as I said, cloned by the end of 79. While Charles was cloning it, I was giving a seminar in Japan.
So at this time, and throughout this, you were still a professor. How did you navigate into that? Because today a lot of profs are also, they work in industry as well. They might be consultants.
Well, that's right.
At that time,
at Harvard, you could be a consultant,
right?
And so, in principle, during that time, you can consult sort of one day or a couple of days a month.
I guess what was your
I was just trying to think, and so by the middle of 1980, we set up a laboratory in Geneva. We've hired a CEO. We have somebody running the company. And in the middle of 81, after the Nobel Prize, I feel that we raised more money for the company at that time.
I'm sure that helps.
We raised $20 million from Shearing Plough, and we raised another $10 million from a British hotel company. What they were doing in investing in it's unclear to me at this time. But what we thought we were offering them. So I'd raised a lot of money for the company. With Phil Sharp, and I remember we were visiting Shearing Plough, and I'm talking about possible investments, and Phil kicks me and says, "Why don't you ask him take both a position in chemistry and biology?" And so I say, "Yes, how about investing $20 million?" We're just talking about $10 million investment, and they in fact do invest $20 million.
Wow. So what were your colleagues who were not doing industry-related things thinking? Because some researchers might prefer to stick purely to academia, and some people.
That's right.
At this time, it was a very novel thing to be founding a company and doing original academic work.
Okay, well, it's a very mixed bag. The physicists had been involved in industry after the war. And that had continued. MIT physics was very closely involved in industry. The chemists generally were involved in the pharmaceutical industry, and a very strong industrial connection there. The biologists up to that point had not been involved in industry. And so some of them were very suspicious of that. The biologists in Europe tended to have industrial connections. They're more likely to have been involved in the pharmaceutical industry or the chemical side or something like that. So Ken Murray had an industrial connection, actually had worked in industry, and they had sent him to college. And it actually made his career possible after an industrial connection. So, but the American biologists were sort of very, very skeptical of this. And as I said, the scientists looking at the venture capitalists were very skeptical of it. And I did not really expect to get deeply involved. But I found myself, because I was elected head of the scientific board, I found myself involved in the money raising, involved in other aspects of the company, to such an extent that in the middle of 81, I could essentially say, the company is moving too slowly, I should run it. And I say to the board of directors, I want to run the company. And they say, no, you can't. And I say, okay, I'll leave. And they say, okay, you can run the company. So I then become CEO of the company over the first CEO we had, who then about a year later left and became CEO of a large drug company.
Did you interact with folks at Genentech at this time? Were you
actually, our interaction with Genentech was a very amusing one, because it's actually on the sort of first meeting we have as a board of the company. The venture capitalists invite Bob Swanson to come because
what was his role at Genentech? He
was
the
CEO.
CEO at that time.
CEO.
Yes,
that's right. So I invite Bob Swanson to come, and I say nothing doing. He's there in the hotel. I say, no, you cannot come to our meeting. You're a competing company.
You're competing, yeah.
I mean, of course,
right.
How can you possibly come and sit on our board and be part of it?
That's very interesting.
So, very
and did you enjoy that whole process of being at Biogen?
Well, I enjoyed the process. Both, I found I enjoyed every aspect of it. That's one of the reasons I stepped forward to say I'd like to be CEO. I thought the company was going too slowly. I found I enjoyed a whole range of things that were novel to me at that time, ranging from everything from the patent law and the question of patenting to doing the investment deals, raising the money, talking up the company, talking up the future. And then when I became CEO, I thought, well, I should read up on being CEO. There must be books of how to be a CEO. And then as soon as you do that, you discover there's no pattern.
There's not 6 papers you could read of how to be a CEO.
Well, it turned out to be nothing you could read that's relevant to it. It's all sorts of people. It requires certain things. It requires a drive to do something. The entrepreneurial drive is actually a sort of drive to make something, again, like the science or like the art, make something new happen. In this case, you were trying to make something new happen in the world. And you're willing to do everything needed to make it happen. The excitement actually in new companies is that it's all new and you're trying to make something novel in the world. Your job as CEO of a new company can be everything from sweeping the floor to, or ordering the things, to making sure that happens, to making sure there's enough money. You're there to make sure that everything else happens.
Mm-hmm. So operations, like handling directing
operations. You're handling operations. I mean, one side of a company is operations, one side is strategic future, one side is raising money for it.
Mm-hmm. And then there's the science of
and there's also the science. You're not actually, in one level as CEO, you're not actually dictating any of this. You're trying to make sure that it happens. You are giving a framework for it to happen. Right. I found it sort of natural to do it as a scientist. I could both talk up the science and talk up the promise of science. I did find a crucial difference between running the company and running a laboratory. In running and being a science in the company, in running a laboratory, we generally had the leisure of trying to be sure that our ideas were true before we published them. And so we could wait and redo the experiments. In the company, time is of the essence. We actually don't have the leisure to make sure things are absolutely true before going on to the next step. You have to be willing to shoot a little more from the hip. You have to be willing to try something and decide after you tried it it wasn't a good idea and you should do something else. And that's a crucial aspect of life in the company, and the CEO has to embody it and has to encourage other people to do that, which means that you have to encourage people to try things and make mistakes. You don't punish them for making mistakes. You punish them for being too slow about getting anything done. Because inside the company, you're burning money.
Right.
Now, you burn money in a laboratory also, but generally have a longer leeway.
Right.
Although running a large laboratory, as I said, is good practice for running a company.
Yeah, and in many ways, that practice of sort of industrial scale, which you can get out of a company, academia was lacking at that time in many ways.
Well,
so
that's why academia should lack it. I mean, industrial scale, in a sense, is the role of multiple technicians. Academia is something else again. Laboratory doesn't exist with multiple technicians. A laboratory exists because you're trying to train students. And the question of training students, as I was saying, is a question of trying to lead them to learn how to decide things for themselves. So I can't take a student and have the student do a project in which I tell the student every step of the project.
Yeah.
I can use a technician that way. I can use 10 technicians and do 10 projects.
Right.
That's industrial scale.
Right.
Students are very different. Even postdocs are different. Postdocs also have a continuing process of trying to learn. And you're trying to do, I often view it as the student needs a project that can be done at their skill level. And so that needs a project that they can work on, which is likely to be successful, which they can learn how to do things. The postdoc often picks their own project. It's the first time they actually have the problem of, how do I really decide I want to do something? And then you can help them around the edges in that. But again, they have to figure out how to do the project. And then become a faculty member. You have the job of picking other people's projects.
Yeah. Great. So this, working in Biogen, working in all sorts of areas, starting as a theoretical physicist, becoming, a leading molecular biologist, and then founding a company. You've thought about lots of different areas of things, and I assume you were interested in other areas outside of science, such as, now you've been an artist for many years. So could you comment, I guess, on the relationship of other fields to science, and including art, and how all these parts sort of fit into the whole?
Well, I'm not sure I have a vision of how things fit into the whole. I do take the attitude people should follow their interests, and so that you should change. I just heard he had a student who has a Nobel Prize. I just heard him talk, and he said firmly, I try to change what I do every 10 years.
That's a good way to be.
He once did recombination. He's now doing microglia in the brain.
Oh, yeah. He's doing neuroscience. Okay. Wow.
Now he's doing neuroscience. He once did something entirely. The Nobel Prize was something different. They're now doing neuroscience. Very much a sort of, I try to change entirely. I think in some ways, when I look at my career, I think I roughly changed what I was doing every 8 years or so. I spent about 8 years in theoretical physics.
Yeah.
I spent about 8 years in protein and DNA and gene expression. I spent about another 8 years in sequencing and recombinant DNA. when I went back to university, I worked in early evolution for about, I came back in 85, another 15 years. We tried also to do something in neurobiology in that time.
And you also did you did some bioinformatics too.
We did some bioinformatics on
using
sequence
analysis.
We was in trying to do sequence analysis in the early days. You began to get some comparative sequences, and you began to look for things. And in fact, of course, we were very much involved in the question of, can we sequence genes that are conserved between animals and bacteria, and do they have an interesting intron-exon pattern, and can we learn something from that?
Right.
We were deeply involved in that in the sequencing world.
So advice you would give is to change up what we do.
Well, that's right. I think changing is always good. People tend to feel that people unfortunately think they've been trained to do something, and they must be doing the thing of training.
You hear people say, "Oh, I'm X by training," and then they stay and do that for their whole life, and that's one way to be. But it's really exciting when people can just pivot around.
That's right.
And like Crick in his autobiography talks about, like you don't, if the field has a clear name and a curriculum, then that's not a good field to be in.
Well, that's right.
Has that been a way?
That's right. Fields which don't yet exist are the best ones to be in.
There were no departments of molecular biology, in the time when all this.
That's right. The time we're doing molecular two aspects of that.
Department of biophysics, and
that's right. Well, we at least called it molecular biology. In some ways, it's horrifying to realize how the science has spread. I mean, science does roughly doubles every 10 years, so that by the time you've gone 60 years in, there's 64 times more science, whatever. And each field, when I worked in molecular biology, each of those fields that I talk about, protein synthesis and control, each of those fields is now grown and subdivided. I worked on ribosomes. Well, ribosomes by now are an entire field of study.
Yeah.
tRNA would be an entire field of study. There'd be people who specialize just in that.
Yeah. People specialize in one tRNA modification.
That's
do
right.
their whole postdoc and PhD on one modification.
That's right.
Yeah.
That's right. So it's that's why one advice is always, I totally agree with Francis, find new fields, find fields which people don't know much.
And maybe not to be like, what do you, I mean, you hear people saying, oh, I don't have a background in X, but what's the point of thinking that way when there's so many exciting things happening?
That's
you
right.
could learn.
That's right. First of all, you can always learn, even in a well-adjusted field, you can learn it even a complicated field in a year or so. And a new field, you can generally learn very quickly because there is nothing to learn.
Yeah.
There's just a gap in one's knowledge there, that something to be looked at. I retired from the university. I thought I'd do venture capital, and I found myself doing art. I've been doing the art for the last 25 years. I fell into the art. At that time, I'd used a camera all my life in one form or another, generally to just record things around the world, as sort of the journalistic use of photography. Although as a child, I had a dark room and did enlarging and developing and that whole range of things. Back in 2000, I had a 2-megapixel camera, one of the first digital cameras. Had a great big lens. It's a big thing to carry around. And I was traveling and made all sorts of pictures in Switzerland. Oh, in Sicily, actually, it wasn't Switzerland, it was in Sicily. And everybody said you can make little postcard-sized pictures from your 2-megapixel camera. And I discovered, since I was most happily using the computer, I discovered I could use Photoshop to blow the pictures up to a much larger size and adding pixels, get images that were quite good, and discovered I could make first 8 by 10 and then 2 by 3 foot and then 4 by 6 foot images from a 2-megapixel camera. That image, by the way, is made with a 2-megapixel camera.
Wow.
So...
What year is this from?
That's about 2004.
Wow.
What is this?
The original image, that image of 3 doors, I'm actually walking through the new wing of the Thyssen Museum in Madrid on a architectural tour. It's a wing they just made for his wife's collection of old masters, and the walls are actually that color, a sort of orange. As I'm walking through, I see this, they haven't hung any pictures yet, I see this series of 3 doors, and I take that photograph. As I said, it's done with a 2 megapixel camera. It isn't sharp, but of course no digital image is actually sharp. What actually happens is your eye completes the image. And so you see a certain amount of texture that your eye sort of interprets as like oil painting or something. And you find a perfectly natural image. And everybody said, you can't do this. And I was saying, look at these images and saying, no, it's an art form. I should do something with it. And so that around that time, early 2000s, I began to play with it as an art form. That's actually...
So what about art really appeals to you? It seems like, you're interested in novelty. And
well,
is there something
as
about
an art
that?
form, it's really that I'm creating a novel image that on this scale, I think has an emotional impact. It has an emotional impact on me, and so I think it has an emotional impact on other people, and so I began to do it as an art form. And so I got this studio we're sitting in and got a large printer, began to print art here, work on the art here on the computer. I use the computer as a tool, and essentially as a laboratory tool. I sort of twiddle the dial and play with the image. Originally, I just photographed fragments of the world, some things that become abstract because they're close-ups of elements of the world. I then photographed the ballet dancers in rehearsal. I photographed a laboratory in Poland, not a laboratory, a industrial museum in Poland, and we did a big display of those images in Poland, and we did a book of the images of the ballet. And after photographing the ballet dancers, which are very beautiful photographs, I began to make abstract images of the ballet dancers, and then spent a year playing with abstract images based on the ballet dancers, and then decided that those were interesting abstracts, and they were all biological, and I should do a geometric abstracts. And so I spent a year doing abstract images of squares, and then a year doing abstract images based on triangles, and another year doing abstract images based on lines. They're not here, they're in the other room. And some examples.
There's lots of paintings around here where you can't all see them, but we'll go around.
That's right. Then after doing that, I came back and I spent 2 years or so doing black and white images, and then went into violet color. Spent years doing violet color images.
What about abstract art particularly interests you?
Well, what I do is a form of abstract art. At the moment, it's done by photographing generally the iPhone and taking multiple photographs and superimposing them and forcing the images, forcing the layers to interact and creating novel colors and novel patterns that way. And I'm not interested in the art as representational of the world. I'm interested in the art in terms of an image that I feel carries an emotional impact or has some feeling of beauty. So that it's abstract in that form, although it can be a little element of the world. Right over there, we have a seascape that's been treated in a way that makes it both real and abstract.
Oh, very interesting. So you like the aspect, I guess, of how it makes the viewer feel, basically.
Yeah, that's right.
Do you feel that way about your science at all, too?
No, the impulse in science is also to create something new. And our test for new has to be new and true, and our test for truth is experiment. For a theorist, the test for truth is that other people agree that the argument has some sort of validity or the deduction is true. in art, I find the thirst to do art is that same thirst to create something new. Constant fascination with, I've done it before. It's no longer interesting. I don't want to do whatever that is. I can find myself falling into, I've done that before and I do it again, but I'm generally trying to find something that strikes me as interesting and new and different.
Who are some people or movements that have influenced you in terms of how you think about things scientifically and artistically?
Oh, I can't even answer that. Probably,
Are there any
I do it all internally. It's really.
Could you point to any scientists?
I am self-driven that way. As you read science when you're young, you find wonderful scientists, and as you get older, you find scientists with feet of clay. Nothing is quite as good as it seemed to be. It's hard to know.
What about in terms of on the artistic side, maybe music, film, anything on those fronts that particularly resonate with you?
Well, I tend to listen to a lot of opera.
Okay.
I enjoy going to the opera. On the other hand, I don't make a deep investment in it. I'm sort of not deeply interested. I enjoy the music, enjoy the singing.
Are you interested in music though?
I'm not. I played instruments at one time, but I never became deeply interested in music. So, and these years, of course, I was going to say I just started going to the opera again after having stopped during COVID. So, high on my attention to my plans for the fall.
Yeah, and so I guess what are some pieces of advice you'd give?
My pieces of advice are of course always to find things you're interested in. I realize I tend to be very self-motivated, and so you see, it's like Jim was a friend, got me involved back in biology. My abilities in biology and his are quite different. Jim says Jim is not a good experimenter, but Jim was not a good experimenter, but had generally a clear concept of what the experiment should be. I mean, some people, I have good hands, but I enjoy experimenting all the time. But I also have a concept of experiments.
And it seems you've always had an appreciation for theory and its importance.
But I think that's right. Well, I went off into theoretical physics, mathematics and theoretical physics. In the biology, but on the hand, in turning the biology was entirely an experimental field. In those years especially, there was no sort of theoretical biology. There's now, I was going to say, we have a greater ability to do things computationally. And we begin to have very powerful computational tools, which we don't really understand. The power of the machine learning is that it can see patterns in things, and we can feed the computer a variety of things and have it learn from those. Mm-hmm. And I'm just blocking his name, unfortunately. I just heard a lovely talk by the guy who did AlphaFold. It was
John Jumper?
Yeah, Jumper. And he was speaking up, yes, we can train the machine to do all sorts of things, but the important thing is to have a test set that's independent of the training set and really verify that things actually work. Yeah. And I thought that's a wonderful statement because, of course, that's exactly what we don't have with the large language models. We're using the computer in all sorts of ways without a test set and with no verification that anything is right. Yeah. So that there's a use in science with test sets and trying to really see that the computer is seeing some pattern in the world that we can verify as it's being seen correctly, reasonably verify it's being seen reasonably correctly, and use it to try to predict other things. And this actually becomes very powerful. People try to use it to predict experimental patterns, to suggest experiments that test things.
Yeah, so in your the paper you wrote in 91 about the paradigm shift, molecular biology is dead, long live molecular biology. You sort of predict that or advise biologists to become computer literate. And I think in that paper, there's an interesting thing you say, which is that the pattern, the process by which we'll do biology will change in the sense that you'll start with a theory and then you'll make predictions, and then at the very end, you'll have an experimental validation. I think it's interesting how much that's actually played out, especially now in machine learning, because I work in this ML for bio field, and so what we often do is you have a model which makes some prediction about a sequence, about a property of it, and then you get the sequences. Once you get them, the real thing that will make a difference of whether your paper is in Nature or not is if you can take that sequence and actually do some experimental process to it, and then see if that matched your model.
That's right.
And really, that's the end of the pipeline, is this experiment. So I think it's interesting how that's played out, kind of in accordance with like what you wrote about in the early 90s.
All I can say is I'm delighted.
You've been interested in theory. What do you think should the role of theory be in biology?
That's a very big question, because ultimately we want understanding. And our notion of understanding is that we have a conception of who does what to whom. Sort of classic vision. Understanding is not just a list of what happens in the world. It's a way of having a strong enough theoretical notion of causation that we can predict how the world behaves and predict how changes affect the world. And that's the whole theoretical issue of causation.
So theory is a way that we can understand, and in particular, it's understanding the causality.
That's right. It's understanding through prediction.
Yeah.
And prediction is not, yes, I can predict the sun's going to rise tomorrow. That's not a terribly useful prediction, but it is a prediction. Newtonian theory of gravitation is a deeper prediction that the sun is going to rise tomorrow.
You often hear people, especially like from other fields, maybe physics or computer science, that think of biology as not being a theoretical rich science, and that's true to a large for lots of history. It's been that way, but if you look at a lot of what's the big discovery or the big paradigm shifts in biology, some of them, apart from DNA sequencing and the repressor and other such things, is theory has been important. Like Charles Darwin's theory. Charles Darwin didn't do any experiments, right?
He did collect. He collected the finches. Let's be honest. He was out there for 2 years on the
That's true.
on the boat, but working very hard,
true. Yeah.
and bringing back all sorts of animals and this sort of thing. And so
he collected data.
People did not do experiments. The dangerous way of saying things. Charles Darwin does observe the world and creates a theory to try to explain the pattern that he sees. And these days we can do experiments. We now have much more powerful techniques than were available to him. We can do experiments that see the evolution in action.
Right.
And we can
like in vitro evolution and sea
We
lice.
can do, that's right. We can do bacterial evolution fast enough. We can do certain types of animal evolution fast enough, but much more rarely. But small animals, fruit flies, we can do a certain amount of evolution. Bacteria is very easy to do the evolution. So today we can do experiments fully to sort of work out, to show the details of the theory. But our hope ultimately is to find theories in biology that will give us have that understanding, sufficient causal understanding of the biology.
So really, it's about understanding, and I see, and in particular, like for example, when you worked out the sequence of the repressor's target, I find it rather interesting because that was, a case of sort of like, this is a sequence that controls a gene regulatory pathway. And now we've moved forward.
Well, that's right. Doing it originally, you hope to learn something from that.
Right.
Now, we realize we learn compared to our knowledge of how proteins interact with DNA even, that was a very primitive little piece of knowledge, and there's a much more complicated detailed knowledge that's now available.
So going back to Alpha Genome, for example, which recently came out from DeepMind, you've got these huge regulatory code models that are trained on, really the entire human genome, but also thousands of functional genomic tracks, and can make very accurate predictions about.
Of course, now I'm going to ask you, what's the test set?
Yeah,
I mean, it is one of the problems of training on the human genome.
Right. Well, that's a good point.
There's a great danger when you train on everything, and then it isn't clear that you actually have any power.
Yeah, I see that. Yeah, but also if you could get a model to predict a specific assay, and then you test it on a different assay that's related but not identical. We've done things like that, and it's been quite effective, I think, for validating a lot of stuff. But I totally agree that it really comes down to what is your test set and sort of what is the ground truth also.
That's right. In terms of using a program to predict experiments that you're going to do to find out whether or not your predictions are right.
Right.
That I think is excellent.
Yeah.
It's an excellent approach. The problem when you use the program and say, "Oh, the outcome of the program is true," that's another issue.
Yes, totally. What are you excited about, I guess, in the future of biology?
Well, I don't really think about biology these days. That's the trouble. I mean, I'm sort of I tend to watch it go by. I mean, the things that I'd really like to know would be things like the real nature of memory, how the brain is ultimately put together in a deep way. We get also little glimpses of things, but we don't really get anything very good.
How the brain works is something that seems way less understood. I'm not a neuroscientist, but it seems like that area is.
That's right. There's all sorts of little pieces of things that are quite lovely, and you can see memory patterns in the brain, but that doesn't tell you anything about how do you actually recover a memory, or how are different parts of the brain working and talking to each other.
Mm. Yeah, no, it is. And I think I mean, in terms of like theory of understanding the brain, I think the neural network field, which was pioneered by Jeff Hinton at the University of Toronto, he and his group really, I mean, apart from being very useful for making predictions about, everything from text to images to now genes, has been also probably a potentially useful way to understand maybe how the brain works. Because, with Hinton, it seems like he was quite motivated on understanding how the brain works, this sort of basic question. Rather than saying, I want to invent a fancy, cool method, he was interested in a scientific question of how does the brain work? Or how does learning work, I should say. Mm-hmm. And so I think, getting back to this like advice for scientists, do you think that's a sort of being guided by questions? Is that something you would say? Because it seems like you could be guided by making a fancy, cool method or being guided by questions. You invented DNA sequencing because you had a question about how the repressor binds a target. That's really the...
Yeah, let's comment about that, because, Fred Sanger actually is an interesting example. He won the Nobel Prize twice. Yeah. He was a man guided by methods. Yeah. He thought the most useful thing was to invent methods to answer problems. And so the original problem was how can we sequence proteins? And his first Nobel Prize was actually for sequencing a protein, doing a first protein sequence. And then he worked on sequencing RNA, then he worked on sequencing DNA, got a second Nobel Prize for DNA sequencing. I tended to be question-oriented, not method-oriented. Yeah. And so, as I've described to you, I describe my life as questions. How does DNA do anything? How is DNA controlled? And the thing I'm most proud of is actually the isolation of the repressor. It was the hardest thing in the sense we did. It was distinctly a question, a problem with a question, and it was very difficult and ultimately surprising. We went down surprisingly wrong paths in the process and finally went down the path that got us to the answer. And the DNA sequencing, which has had the biggest impulse in the world, actually is a method rather than a question. And that discovery was by accident, and been very nice. And the world has changed profoundly by the DNA sequencing discoveries.
Yeah.
And I'm involved now as a consultant in a company that wants to sequence babies.
Mm.
And there's a whole sort of side of should we sequence everybody? And I think ultimately, if we had a rational medical pattern in this country, we would sequence everybody. That you would learn something from your sequence that would be of medical use to you.
Do you think everyone should get their genome sequenced?
I think ultimately we should sequence everybody.
Uh-huh.
Only about, when you sequence babies, it turns out about 2% of the time there is something seriously wrong that you can do something about. And about 10% of the time, you actually see with our current knowledge in the sequence something that's going to be relevant in their life, something that's likely to be a heart problem or a disease problem later. But 90% of the time you have a DNA sequence, nothing's not going to learn anything from it.
Right.
Eventually, we'll learn more.
Yeah. And ultimately, a lot of complex human traits, perhaps, most complex human traits are polygenic or they're caused by multiple mutations. So really, just reading the sequence alone is not enough. You have to have a regulatory code model that can interpret variants and also interactions between variants, which is, I think, something
it's actually right. I'm involved, the company, peripherally involved these days, the company Myriad, which analyzes breast cancer genes that predispose one to breast cancer. And they actually test for about 6 genes, and then they test all around the genome to see controlling variants. And the variants change the effect of the genes by, up to factors of 5. And major effects on whether they predict whether somebody's going to have a case of cancer connected to the gene.
So that is very powerful. Ultimately, is we should move towards a world where people a sequence-centric healthcare, perhaps, in the sense of.
Well, that's right. The only reason we don't sequence babies because the company that is paying for the sequencing isn't going to take care of them later. Doesn't care.
I see. So there's an ethical question of what happens once you have the knowledge that.
No, well, that's right. There is an ethical. There's 2 issues. There's just the economic issue at the moment. There's no source in the country that actually has an interest in the person's health throughout their life. A great chunk of our medicine is paid for by Medicare. It takes care of the older people. It pays for about 1/3 of the cost of all the medicine. Another 1/3 is paid by the VA, and about 1/3 is paid by insurance. But the insurance covers all the young people and makes all the fuss about how we should be doing medicine. And so there's nobody, a sort of question of, well, what is the medical care that really you should have that tries to look at you when you're young and predict are there going to be problems and is there to take care of you when you have the problems? There's no place for that in our medicine. And then there's an interesting question that gets involved in sequencing. About half the people like to have their sequence done. About half the people don't want to have their sequence done. It's always been an interesting aspect of this. People worry that something might be learned about them to be a problem. You think, I don't think there's actually ethical questions. There are practical questions. Is there going to be insurance problems if I have my sequence done? The answer actually is that the laws against the insurance companies using genetic information. It actually is a piece of law in Congress that got passed across this period. The insurance companies still use genetic information in determining life insurance. Because they charge women and men differently. Because women and men have different life expectancies. We allow the insurance companies to charge on that basis. It isn't clear we should, but we allow them to. But we don't let them charge. We don't let the health insurance companies charge pointedly on gene defect. And what other ethical issues do you think?
Well, there's sort of the question of how to act once you have information. I think that's the broader question, because there's in ethics, when you the revelation of data implies some decision. Like if you have knowledge, the A versus B, if you have knowledge that someone is suffering versus if you don't. If you have knowledge that they're suffering, then there's an ethical imperative to help them. So likewise, if you have knowledge of someone's sequence versus if you don't, there's the things should I do this or that or not? And insurance companies, for example, would in principle, I'm not commenting on whether it's right or wrong. I'm just saying they would leverage as much data as they can get, and not just them, but anybody would, any company that's for profit or anything. So really, it's about what do we learn about from sequence, and how much should we act on it, right?
Well, that's right. But of course, it's also the question of how much do you learn from your sequence? Do you want to act on it? There was a music case in the case of Huntington's. A woman who was the most involved in actually searching out and identifying the Huntington's gene never got herself tested. She eventually came down and died of Huntington's.
Oh no.
On the other hand, if you know you have carriers of the gene, that knowledge enables people to now search for treatments.
Right.
And treatments that can be applied early. So the knowledge is ultimately very useful to society. Yeah. And even though you see the knowledge of the person seems to be, I have a defect that is going to be, I have a problem that's going to seriously impinge on me.
Yeah. So ultimately, it seems that there may be a good case to be made that there's more benefit than harm that could arise from the right people knowing about our sequence.
In certain ways, I suppose, I think the attitude, there is no harm. There is benefit, or you don't do anything.
Right.
But there's no particular harm involved. There's harm involved if you allow the sequence. There's no knowledge to be used by an insurance company against you. But as I said, we pass laws to try to prevent that.
And also the theories we currently have about the influence of genes on behavior, which is a controversy that we can get into another day, is like our models of that are not so great anyway. So,
that's right.
There's not really much you could learn about behavior from genes. So it doesn't really matter.
Well, that's right. There's lots of nonsense about it.
Yes.
So it's not. One side of that is unfortunately the entire side of racial bias gets backed up by bad motivated people, the physical anthropologists, who desperately try to find genes doing bad things to people, or people with bad races with bad genes and things like that. And most of that's just entire nonsense.
Mm-hmm. Yeah. So hopefully the good will prevail, and we'll move towards a better world where healthcare is improved in part because of sequence and sequencing, and sort of our understanding not only of the world but of ourselves, and life itself, and how life arose and how life evolved, and where we might head to next. So with that, I think I'd like to thank you, Dr. Gilbert, for your time today and for this very exciting conversation. And that will be it.
Thank you.
Thank you.